So I think we begin the second half of this session, which I am going to chair. And I would like to welcome Patric Baumann, who is a PhD student of Professor Wolfgang Meier of the University of Basel. And he is going to talk to us about polymeric nanovesicles. Please.
Thank you very much for a short and kind introduction. I'm welcome that still a few people left here. I'm the last person of the NRP 62 who will present today. And I'm talking about polymeric nanovesicle generating reactive oxygen species for efficient cancer therapy. First of all, I would like to go on the expression polymeric nanovesicles. We in our group of Professor Wolfgang Meier, we work with so-called polymeric nanovesicles, which we think they should look like this. They consist of a polymeric membrane, which should somehow mimic artificial membrane, and has an aqueous core where we can encapsulate different, in our case, mainly bioactive molecules. Additionally, we try to permeabilize this membrane either by incorporating of membrane proteins, or we just use the ability of the penetration of different molecules through this membrane. We have worked on different applications like biosensor, when we have an enzyme in it to detect molecules, on drug delivery, as we have also a delivery system. Nanoreactors we can produce in this reactor also kind of molecules, also with the help of enzymes, or we can use them as artificial organelles, as these are rather stable self-assembly vesicles, which we can also give to cells. The other topic, or what's my topic about, is the photodynamic therapy. It's a new approach for cancer therapy, and the aim is that the patient doesn't have to suffer so much anymore. The idea behind this is that we have 3 different components. So one is light, one is a photosensitizer, and the third one is the oxygen. So what I have done, I have formulated kind of the photosensitizer, which we then can illuminate with light. This will then go to an excited stage, and the energy can be transformed from here to the oxygen by then creating radicals or singlet oxygen, which then, when in cells, can lead to cellular toxicity. In this scheme, you see how it could look like in future, that you apply your nanovesicle solution. It should find the cancer somehow. If you irradiate it with light, then the patient should be treated. The advantage over classical cancer therapy methods is that it's not invasive, it's no scared, it's targeted, it's not active targeted of the nanovesicles, it's targeted according to the light you can apply to the cancer and the photosensitizer which is there. It's also a rather fast treatment compared to chemotherapy. It's repeatable and it should have low side effects. The nanoreactor I have used for my studies consists of a polydimethyl oxazolene polydimethyl siloxane polydimethyl oxazolene polymer, which forms this round stuff, which looks under electron microscope rather weird, as they're normally a bit collapsed structures. So we always compare our vesicles with liposomes. So because the liposomes, they're not so stable, and we believe that our polymeric system and also other polymeric systems have a higher stability and therefore are more suitable for drug delivery or for other application. The good thing in my case, I didn't have to incorporate any membrane protein to get the reactive oxygen released. Our membrane is permeable for RS and therefore I can just entrap the photosensitizer in the vesicles and then illuminate it. Additionally, if you would give the photosensitizer only by itself, you would dilute it, but we have it encapsulated in vesicles, and therefore we always have a high concentration, like a bomb, when we can illuminate, and then it will release reactive oxygen species. Here is the photosensitizer, the rose Bengal. It's a natural compound which has a quite high efficiency to turn light into reactive oxygen species. But we first, when we did the first trial, we recognized that we had a problem that this compound is somehow binding with our polymeric membrane, and therefore we conjugated it with BSA to get it more hydrophobic and therefore not interacting with the membrane, but only have it inside. With this, we also lost kind of activity of the rose Bengal. As you see here, we have done some spin trap experiments and detected the radicals produced by illuminating with normal bulb light. And you see with free rose Bengal, you have quite fast increase of the radical detected, but with rose Bengal BSA, you see that you have a little bit less production, but on the other hand, we can play with the light dose, and you also can play with the concentration of the polymeric nanovesicles to increase then the free radicals for the treatment. What we have seen that by conjugating the rose Bengal to BSA, we have a shift of the wavelength, of the absorbance wavelengths, but on the other hand, we have separated our nanovesicles, which are generated in a self-assembly process from the not encapsulated rose Bengal BSA, and we see that we have the rose Bengal BSA in this fraction, the free one, but we don't have any free rose Bengal BSA in a first fraction. So we only really have it inside the vesicles. For the characterization of the vesicles, we have a diameter of around 200 nanometer, which is suitable for the EPR effect, so would be kind of a passive targeting. We can also reduce this size down to 50 nanometers, and we have an encapsulation efficiency of our conjugate of about 13%. To be sure that our nanovesicles is suitable for cells and not toxic by itself, we did some dark toxicity measurements because we aim that our vesicles are toxic by illuminating, but without light, the vesicles are rather not toxic and only have, we have about 90% viability up to a rather high concentration. To be sure, I worked with a concentration of 100 microgram per milliliter for the following experiments. To see if the singlet oxygen which are generated by light are coming out of the nanovesicles, we also have done this EPR, the same experiment as before. And we see we have around the same trend as with the free rose Bengal BSA. So we do not have a limitation according to the encapsulation of our vesicles. And the next step we have done is to test is we brought our nanovesicles in contact with cells, with HeLa cells, and then we tried to measure the free radical concentration inside the cells we could generate. Therefore, we used this compound, which can be activated by esterases only in cells, that you can measure the spin trap in the cells. Therefore, we have two things we can read out of that. First of all, that the signal is with the irradiation time increasing, and secondly, that the vesicles have to be inside the cells, as this compound is only active inside the cells. Additionally, we have done in parallel a study by illuminating the cells with uptaken nanovesicles, and we see that we have a light-dependent decrease in viability of our cells. In the next step, we have looked under the microscope what happened there, and what we see here, we have in green the cell membrane, we have the nucleus, and we have in red, we have our vesicles. We don't know exactly how the vesicles enter the cells or where they are at the end, but we just know that they are at least within the cells. And if we illuminate it with a specific wavelength, with a laser light of 543 nanometers, we can see that afterward we have kind of a bleb formation, which is an indication for apoptosis. And this we could do over a quite short time period, and also with a quite low light dose. And the astonishing thing which we saw afterwards when we zoomed out again, that in this region where the bleb formation appeared with the light, first of all, we don't know what has happened with our photosensitizer in here. Maybe it's bleached or anything, but outside the cells they were not affected from this treatment. So outside we don't have any bleb formation, and therefore only where we have the nanoreactors, where we have the light, and where oxygen is, the treatment is happening. So therefore I hope I could somehow convince you that we have produced stable and reactive oxygen permeable nanoreactors which can be uptaken by cells and produce inside the cells reactive oxygen species. Additionally, they are light triggered and only active when light is applied, and therefore we have a kind of a controlled cellular toxicity with this combination of the photosensitizer, the light, and the oxygen. Therefore, at the end of my talk, I greatly acknowledge the NRP 62, which made it possible to do my PhD here in Basel. Additionally, I have to thank different people who helped also during the work and everything like this. Yeah, and you for your attention.
Thank you very much, Mr. Baumann, for your interesting presentation. There must be questions today. Yes, please. Please do identify yourself.
Yeah, sure. Zbigniew Brzostka, Technical University of Warsaw in Poland. I have two short questions. First, you show the power of a laser. This is a surface measure and surface or measure in the tissue. The first question, this was in joule to the square centimeter. It was measured at the surface power given by laser or in somewhere in the tissue depth?
Surface, because
surface.
We only had tissue culture. We didn't really have tissue by itself.
Okay.
We just have a monolayer of cells.
Okay.
So
Another short question is whether these vesicles are targeted to the certain kind of cell, so it means the cancer cell, or these are just a nanovesicle without any targeting molecules?
So targeted can be when EPR effect is applicable. The ARS which are produced is not specific for any cancer. You normally should be able to use it for any cancer, as they just killing the cells and they don't block any mechanism or anything like this.
Okay, thank you very much.
More questions? I have a question. You were talking about blebs.
Yes.
And how do you know? I mean, you showed only light micrographs. Did you do any electron microscopy to show these blebs?
We haven't done that yet.
How do you know then?
The blebs form.
Yeah.
Yeah, we see it.
What do you call bleb?
This.
Oh, I
this,
see. Okay.
Blebs which are forming.
I agree. Thank you. Are there other questions? Last question, yes?
Just
Please.
a comment. I think that
Please identify yourself.
Oren Regev from Israel. I think that your collapsed liposomes or vesicles, the terminology that you used, is due to the fact that you took the
water out, I guess.
yes,
yeah.
you took it in room temperature, which it was dry, and therefore
Yeah, we have also done light scattering experiments, and there we saw that we have the radius of hydration and radius of gyration are in the same range, and therefore we know that we have a spherical structure, but in microscopy it's nice to show, but yeah,
All right.
it looks collapsed, yeah, that's right.
Okay, thank you very much again.